intensity of cancers it's inviting.
Yeah. Aya, can you turn on the, so you turn on the video, but can you share the presentation and so that we
good evening to everyone participating from Sri Lanka, and very good morning to everyone participating from the USA, especially our invited speaker, Dr. Roshan Liyanage, joining with us from the USA. As the head of the Department of Chemistry, University of Peradeniya, I am pleased to welcome all of you in this beautiful evening to the 14th webinar of our Inspire series conducted by the Department of Chemistry. So today's Inspire webinar is kind of a special webinar for us. That one of the main thing is that the invited speaker is also a member of our chemistry family. I'm sure this lecture, especially the webinar, will be a very fruitful one for everyone. Thank you very much, Dr. Liyanage, for accepting our invitation. In spite of your busy schedule. I know you are also very happy to be with us, at least virtually, since this is your place, your department. So without taking much time, I would like to invite Professor Ayanthi Navaratna to introduce you to the audience. Have a pleasant evening, everyone. It's over to you, Professor Ayanthi Liyanage.
Thank you, Professor Manu Devi. I'm very pleased to introduce today's speaker, Dr. Roshan Liyanage, and currently he is the Associate Director of the Analytical Division of Vaxcyte Incorporation, California, USA. As we know actually through the screen, his specialty or the expertise is mainly on vaccine production, but in addition to that, he has expertise on immunotherapy, cell therapy, and gene therapy. So you know these are very novel technologies, and I think they are maybe in practice in US, and we don't even have incorporated some of them even to our curriculum. So we can do that in the future. When it comes to vaccines, I think Dr. Roshan is specialized in bacterial vaccines, and especially pneumonia and for diseases or infections which are antibiotic resistant. And you may know already this is a big issue in Sri Lanka. Even in India, we have antibiotic resistant tuberculosis. That is a big issue. It soon it will come to Sri Lanka also. So I think he is involved with very important investigations, research activities. And you can see that when we get his research discipline, it mainly, I think he did analytical chemistry, but there are no single discipline this day for research, and it's a multidiscipline research. And so he has a combination of biology, and I think for him, chemistry, analytical chemistry, and the biology, they are both are equally important. I think he will explain to us that one soon. And before accepting his new position, I would say new position because I came through the FB that you accepted this new position at Vaxcyte. He worked in several groups like Dendrian Group, Corporation, Bristol Myers, and these are well-known places in USA as his previous employments. Very importantly, Dr. Roshan is currently served as a chairperson of the Biotechnology Advisory Committee of an organization generating biotech talents in Washington, USA. Of course, we have to have talent to do this kind of work. Of course, we in Sri Lanka, we are producing many talented chemists, and we are proud to say that. And in addition to his, we know that he's a real big person for us today, but I still have his sweet memories as an undergraduate. That is our Roshan at that time. Roshan was a very active member in the Buddhist Brotherhood Society. I remember he was actively involved. And also he came to Sri Lanka to participate the NSF program conducted to get the service of our scientists, local Sri Lankan scientists who work abroad. Unfortunately, it didn't work, but he had a very keen interest to participate into this program. I also participated that NSF program. But I think still he has the keen interest of participating in various activities of Sri Lankan things. So I believe, although he lives in US, he has a keen interest of participating chemistry activities, not only Peradeniya, in other places also, as well as to Mother Nature. His motherland that is Sri Lanka as a whole. So I'm very pleased to invite him. So I cordially invite Dr. Roshan to present his findings. Thank you very much.
Thank you very much, Dr. Professor Anthi, and good evening to everybody in Sri Lanka. I think what Professor Anthi said was absolutely right about me, that I have keen interest in Sri Lankan, especially the academic areas where the chemistry and biotechnology can make a difference in people's lives. So I've been trying to do many things, but I think I had to keep trying. So that's how it works in Sri Lanka. Before I actually move on to the presentation, there is a kind of request that I also put some of the slides outlining who I am and how I get there. It's mostly kind of the pathway that I kind of created for myself. I think I would talk about I'm gonna have a couple of slides just to talk about that. That would be kind of my academic path that I created to get where I am, and also to accumulate the experience. So I think I'm going to talk about that at the end of the seminar. Let me. I'm going to share my screen, and okay, let me see. That works. Can you all see my presentation in the presentation mode? Someone tell me if you all can see it.
Roshan, I can see.
Okay, so that's in one screen, right?
Yeah, that's my screen.
Okay, all right. So just wanted to make sure that sometimes this the Zoom or the web portals acts funny. So thank you so much. Again, the topic that I chose here is just the analytical chemistry perspective into frontiers in biotechnology. That's where the industry is headed right now. Like Professor Anthony mentioned, there is no single siloed field presented now as analytical chemistry, physical chemistry, biology, statistic. I mean, in the future, and actually it already has started. Everything to be interdisciplinary. So that's what I believe in. That's what I built. I actually that's what I started believing in. About 10, 15 years ago. So I just realized that, and then I started kind of building on that. So please remember that if you're going to do something, interdisciplinary areas are the ways to go. For example, so like now there is, I mean, most Sri Lankans are familiar with the nanotechnology. Nanotechnology is not a single field; it's an interdisciplinary combination of multiple fields. It's the same way for other fields too. Biotechnology. Biotechnology is not a single field; it's a combination of everything: molecular biology, biochemistry. Chemistry. You name it. So the idea is just what I'm trying to deliver, or the take-home message is try to stay interdisciplinary. If you're not there, just try to get there somehow. So there are multiple ways to do that. So that's what something I'm going to give you guidance on. Even this presentation is not going to anyway going to kind of boil it down what you can do. This is kind of I think of this as a framework where you can just start thinking about it. If you have other questions, if you are unsure about your path, just always reach out, and I'm always willing to help. You can ask Professor Anthony other information. I mean, you can get connected through LinkedIn. LinkedIn is a great source to both learning and networking. That's what matters the most. So here, what I say is opportunities for chemists and biologists. Again, that's not going to narrow it down to just chemists and biologists. So I would just say that's probably my experience. But even from this talk, there are others who can benefit from it. So without kind of further ado, I will start going into the presentation mode. So let me tell you a little bit about the history of the biopharmaceutical industry. So biopharmaceutical industry began in 1982 with the approval of Humulin. That's a insulin analog that was first approved. Since then, industry has grown very, very fast, launching nearly 300 biopharmaceuticals. To give you an idea about in 2018, the global sales of 210 billion was generated from 269 marketed products. So you can see, actually, you can see here that, just trying to see my, okay, hold on. I'm just trying to get my laser pointer. So you can see that the growth was tremendous in field, and in the field. So when you have this kind of growth in the field, if you look at this year, right, the vaccines from Moderna and Pfizer generated so much to add to these numbers. So every time a product comes to the market, there will be, for every product coming into the market, there will be investing 100 products, because when you start with 100 products, you probably may come up with just one product into the market. So the opportunities there is actually it's not just selling it. So opportunities for scientists like us in the making of it. In the making, actually knows a lot of research, development, scaling up, and activities like that generated a lot of job opportunities, as well as more business opportunities if anyone is interested. So the emergence of the regenerative and personalized medicine also has expanded the biopharmaceutical horizon. For example, advancements in immuno-oncology for treating cancer and autoimmune diseases, personalized and precision medicine, also drug development for central nervous system alignments, for example, Alzheimer's, Parkinson's, ADHD, and that area also has, again, the traction into the immunology, because right now most of the disease treatments, I mean, in the past, that it used to be pharmaceutical small molecule drugs. So now small molecule drugs are getting replaced by biologics and large molecule drugs like antibodies, cytokines, and cell therapies, gene therapies, and multiple protein and protein drug conjugates drugs. So I can explain a little bit about this in coming slides. As we all know, the vaccines are leading the fight against devastating diseases. For example, we all know that currently the vaccines that we use for the mRNA-based drugs are mRNA molecules encapsulated in lipid nanoparticles. As you can see here, both the current leading two vaccines are from Moderna and Pfizer, are lipid nanoparticle-based vaccines. So this is actually a huge field right now. Actually, in a very steep uptrend in development. So in addition to that, there are multiple vaccine modalities that are being used, including viral vectors, attenuated viruses, DNA vaccines, virus-like particles, subunit protein vaccines, inactivated viruses. All these are vaccines that are actually currently being used even for fight against COVID-19, for prevention, and most of the drugs actually are marketed now, but there are some others like subunit protein vaccines are already going through the approval. So another area that I'm really currently working on is the polysaccharide conjugated vaccines. Usually polysaccharide alone would not give a very good immune response. So it's when we when you have a conjugate the polysaccharides to carrier proteins, these carrier proteins are coming from some of the detoxified version of diphtherial proteins and proteins from multiple bacterial species. We purify that protein and purify the polysaccharides from disease-causing bacteria like Streptococcus pneumoniae and other species. So then what we do, we actually do a conjugate these molecules, these polysaccharide molecules into the protein via special chemistry. So that would actually generate the vaccines against bacteria. So as you can see here, only the polysaccharide conjugated to these proteins generate immune reactions. And actually, I have plenty of time. If you if you feel like asking a question, I know that we have 30 minutes after the presentation. But if you feel like you want to ask questions during this presentation, please feel free to interrupt me. I have time to stay a little longer if you want to. And the other area that I expand is explosive growth in emerging regenerative and personalized medicines. These include gene therapy and multiple cellular therapy approaches. Gene therapy, when I started, when I was in graduate school, I heard about gene therapy, but it never became a reality until a couple of years ago. So these advancements in the biology, chemistry, and actually the hardest thing is actually delivering these therapies through into the disease causing areas, like for example, like delivering some biologics or other drugs through the blood-brain barrier has been really challenging. But with the novel development in the delivery technology, especially including we all know that the nanoparticle advancement in the lipid nanoparticles have paved the way for mRNA vaccine to fight these diseases, especially the COVID-19 right now. Since I'm using COVID-19 because it's mostly kind of used terms and used. We all are actually familiar with the disease. And so one of the immunotherapies that I worked for, that was my first industry job right out of graduate school, where I was working on a kind of engineering dendritic cells in our immune system to fight against prostate cancer. So there is a treatment called Provenge, exists today for treating prostate cancer. So, and there are from that point, there have been multiple approaches came into the clinic with regional in the region. It's actually included in the regenerative medicine area, because the first thing is that usually our immune system do not attack cancer cells, because the cancer cells are our own system. Our own immune system recognize them as our own cells, so they won't attack them, because it's just like our own attacking as like autoimmune diseases, right? So because of that, we have to engineer these cells, these immune cells like T cells and other immune cells to so they can identify these as a foreign species. They are not our own system. That way, that we can, with our immune system, can go ahead and attack them to treat this cancer and other diseases. So one of the most advanced form of cellular therapy is called CAR T cell therapy. It's called chimeric antigen receptor T cell therapy. What we do there, so it's actually the form of harnessing the power of immune system to fight cancer. So this is more a personalized approach to treat cancer. This approach is widely used for multiple blood cancers, but also being investigated for treating solid cancers as well. So what happens here is actually the patients, we get the patient's blood, and also selectively separate and selectively separate the T cells from patient's blood, and also genetically modify by adding a gene to the T cells to create these molecules called chimeric antigen receptors, where those receptors are specific for each cancer. As you can see here, this CAR a specific receptor is added to these T cells. All these receptors are, we can create these CARs specific to each type of cancer. That's when we start recognizing cancer as a disease, not disease, cancer as a something that it should fight. So starting from that, when we get this kind of, actually what we do, then after gene transfer, we expand this ex vivo. So in a more of like a bioreactor, so a small bioreactor, we expand them and put it back to patient cells. So then once we put back these activated CAR T cells into patient's body, they start actually fighting and killing the cancer cells. The biggest issue in implementing these technologies is especially in low income, low income settings, is that first of all, the kind of development of these costs a lot of money. And once we develop it, even after developing, we have to, for each drug product lot is just one lot. So this one lot, if you think about that, we had to do like, just for the administration from taking the blood to putting it back, we have to perform, actually, I would say like, 30 different tests. It would be like, much more than 30 different tests. These tests are very expensive tests, like, identifying we need to identify if these CAR Ts are formed, and to make sure that, there has been no adventitious agents inserted during the process. That would be like testing for bioburden, endotoxins. There are multiple very expensive tests that need to be done before we give it back to the patient. So that's why these technologies are very expensive, but there are ways that we can bring down the prices of these ones, but it's kind of, those are the researches that are currently going on. Okay, so now, I did not summarize all the the treatment modalities. There are multiple treatments for this. There are antibodies, cytokines, antibody drug conjugates, in addition to what I have mentioned, like all the vaccines, cell therapies, gene therapies, stem cell therapies. There are multiple of these available right now, and also all of these are in continuous development from discovery to market. There are tens of thousands of products that are being currently for all these diseases, all these areas that I biologics areas that I mentioned. So when I during these talks, when I say biologics, it's more biopharmaceutical. Biologics include everything. Biologics include cell and gene therapy. It includes vaccines. It includes all the other antibodies, antibody drug conjugates, cytokines, and all these species and other biologics like insulins. And so all these are included here. So if you can think about, when we try to implement these drugs in these treatments, therapies, it will be much more complex than a small molecule pharmaceuticals. If you think about a paracetamol. Right, paracetamol is like a very small molecule. The complexity is very simple. The chemistry of manufacturing is well established, and and if you compare like a tiny small paracetamol molecule to a very large protein or like cell product, if you can imagine the complexity coming from the heterogeneous product like proteins, cell therapies, gene therapies, because they are highly heterogeneous and require multiple testings, and also make sure that keeping like in a small chemical. As chemists, we all know that it's not the chemical should be happy with a happy medium, but keeping the molecules like larger molecules like cells, the DNA, viral vectors, or for that matter, just antibodies, antibody drug conjugates, and any proteins, it would be really tough to keep them happy, because we all know that these molecules are large, heterogeneous, and there are multiple degradation pathways. So, but let me tell you something shared among these therapeutic areas for vaccines, cell therapies, and the one thing, there is one common development pathways for all these. So that's where the interest should come from. So all these for production of proteins, manufacturing proteins, manufacturing gene therapies, cell therapies, it all start with the expression vector. Expression vector is typical vector that you can you normally see. That this is a vector, like could be a bacterial vector or viral vector. You typically use viral vectors for gene delivery. What we do, we actually take a vector and insert the gene into this vector, the gene of interest. If I want to make insulin, if I want to make a particular antibody, if I want to make a particular protein, so anything, you name it. Like, let's say if you have let's say right now, kind of for gene therapy purposes, if you have to people have these diseases, some hereditary diseases, because they are missing certain genes. So we can actually insert that gene into a particular vector, and the vector actually then that vector is included into a cell line. So these cell lines, there are multiple cell lines that we can use for biotechnology applications. This could be mammalian cells, this could be insect cells, and this could be E. coli, E cells. There are multiple cell lines that we can use to get our protein expressed in there. Then this process is called transfection. Once we have our viral vector, expression vector inside the cell line of choice, then we have to, of course, just do a selection. All the cells that we infect with viral vectors would not behave the same. So we have to identify the cells that are actually performing for our expectations. So this could, for that we have to grow these cells that carry the viral vector. We have to grow them in for example, like in 96 well plates. And once we grow them in each well, and each well will be tested for to see if these cells are healthy, if they are producing the proteins that we want to produce, if they stay stable, or if they create some unwanted species. In that way, this is called the selection process. This is called the clone selection, clone expansion and clone selection. That way, we actually identify the number of good high producing clones or cells that are actually can be moved on to the next phase, where we actually expand these cell lines. Once we identified a really good cell, a really good clone that produce these cells, it's called a stable cell line. Then we actually go through something called cell banking. Cell banking means we expand them and create hundreds of vials, maybe hundreds of thousands of vials of that cell line, which is expanded through SEC+ or other expansion modes. Then we actually, once we create that, we just actually keep that as the called master cell bank. So the master cell banks are kept at under cryogenic conditions. We do a lot of testing here, and actually the growth, once we have the master cell bank, I will explain the process later. But up to now, I can tell you the contribution from the biologists, chemists, microbiologists, immunologists, and molecular biologists is enormous. Then also at the same time, the opportunities are endless I mean, if you go here, like you're creating these viral vectors and making sure that these viral vectors, so you had to do multiple testing here. You had to do qPCR, and also to identify this, you know, had to do viable cell densities and other cell health indicating tests. So, I mean, for each kind of cell bank that we generate, we will be doing like, you know, thousands of iterations just to get here. So imagine, like, you know, how many testing that you had to do. Like, you know, for this selection, we had to identify, you know, the titer. We had to measure the concentration of either the proteins, antibody, or whatever we are trying to make here. And there will be multiple testing here. And also we had to make sure before, when we do the cell banking, we had to measure multiple attributes, including the cell health indicating cell viability, protein concentration, and show that all these are free of adventitious agents like mycoplasma, plasma, and other microbial, like bioburden testing. There will be, like, you know, multiple tests that we have to do, but, you know, there will be, like, you know, millions of tests when we bring one product to the market. Once we have this, then we will be kind of assessing this for scalability and beyond. So I mean, in the following slide, I'll be talking about that. You know, I can give you a rough idea about what happens once we develop the cell lines. We actually expand and make a stable cell line, and it goes into the process development. Process development takes place it will take, even if I say 3 to 6 months, this could be sometimes, you know, years. Then we have to do scale up, because, you know, once we have the product identified, the cell line identified, then we go into scaling up. Scale up also takes place in multiple stages, which I will be talking about in the upcoming slides. You know, once we have these early development done, then we have to go into the cGMP manufacturing for clinical applications, going into the phase 1, phase 2, and phase 3 clinical trial, and beyond that would be commercial manufacturing. So I think, you know, this is kind of a outline of what's common for all treatment modalities. Like I said, for vaccines, cell therapies, gene therapies, you know, I mean, there will be variation. However, so this process is pretty, this is kind of the common denominator for all the processes. We'll see. So here is an example of a biopharmaceutical development process. Like I mentioned before, so first start with the molecular biology side, cell line development, and then clone selection, cell banking. And so once we have the cell banking, then we take, like I said before, we create a master cell bank. The master cell bank will be each vial. I mean, we get like maybe like 1,000 vials for the master cell bank. We'll take a vial and create expand that, and also create from that something called working cell bank. So we create, like, you know, expand that single vial and create one again. I mean, thousands of vials from that. Then cryogenically preserve them. Take each vial for each development run. So you can say here that working cell bank will be used in cell culture bioreactor, and so we expand this into larger scale, and then once we have that, then harvest the product just by removal of the cell, and once then we get out the media from that. After centrifuging and removal of cell, we get it's called upstream harvest. So then that goes into to multiple columns purification steps to remove the unwanted species. I can show it in the coming slides. So this also we also add viral clearance steps. And viral clearance step is just like, you know, if the process, we have to make sure that or the product that we make are clear of any adventitious agents. So there will be viral clearance steps. and just to make sure that there is no bioburden in there. And finally, once we have it goes through a concentration and buffer exchange system, finally goes into a formulation. So this is a typical process development for biopharmaceutical, but I will explain this in greater details for each step. Okay, so I think we have time now. So like I said, this working cell bank vial coming from the master cell bank, and we thaw it and to do multiple testings. Like I said here, before it goes into Shake Plus, we make sure that there will be measurement of the viable cell density, protein identity. I mean, it could be protein or other biomolecules. So we do identity testing, endotoxin, and bioburden stability, just to make sure that there is nothing going into when we expand the cells. So at the Shaker Plus stage, this is probably 1 mil stage, then this goes into probably 1 liter or somewhere around that ballpark. For the testing, that we would be viable cell density, protein concentration, nutrient consumption. So this nutrient consumption is actually just for glucose, pyruvate, and anything that would be used in the cell cycle would be, added and monitored, and the pH. Once we have this, then this will be scaled up to the next level. So we make sure that we can get 1 ml to 1 liter, then make sure that we can get it to 100 liter. See, it's called seed fermenter. At this stage, actually, this is a more bioreactor, much larger bioreactor that has more capabilities. We can control oxygen, dissolved oxygen, nutrients, and pH more closely, and the temperature of the reactor. So usually the measurements, what we measure here, viable cell density, protein concentration, nutrient consumption, pH, dissolved oxygen, temperature, mixing speed. All these will be used because all these parameters are critical for the cell health and also the production of the biomolecule of interest. Once we have nailed down the conditions at this scale, so this, all these steps, like going from here to here to here, and from there we'll take multiple, hundreds of iterations going from this to here. And again, like I said, there will be a lot of work needs to be done. And from seed fermenter to the production fermenter. This would be production fermenter could be from could range from maybe 2,000 liters to 20,000 liter and something like that. All these are so this is the production scale. So for at this stage, also same thing, cell viable cell density, protein concentration, nutrient consumption. All these will be monitored and will be optimized because this is at this every stage. That's the stress that these cells go through different because you can see that the surface to volume ratio is always different. How the material flow, the air flow, the liquid flow, and the stress, shear, everything will be different at different stages. So what we have to understand that for each stage, we have to optimize these conditions just to keep the cells happy and keep the protein production at maximum or optimum level. So once we have this one, then we go into the harvesting, which means we once we take this the media out of here and do this. If the proteins are produced inside the cell, we have to do cell disruption. But if the proteins produced excreted into the out of cytoplasm, then into the media, then we have to just separate the cells. And sometimes we have to do a flocculation. That means just kind of separate the proteins and do centrifugation to get rid of cells. And another filtering step is called tangent flow filtration or UFDF, ultrafiltration, diafiltration step. This is just to kind of separate protein from other unwanted species. So now once we have this one, this would be this is called sometimes upstream retention, retentate, or that goes into the next step in the manufacturing process. This is called the downstream process development. It is actually the purification. Purification involves multiple chromatographic steps. So like I said, this ultrafiltrate retentate is coming from the previous step, from the harvesting step, goes into the first column step. First column, the first chromatographic column is called the capture column. So these capture columns, the modes of chromatography could change from affinity chromatography. For antibodies, there are resins called affinity and protein A resins. These actually capture the proteins, but if you don't have affinity columns, then you probably have to use anion exchange, cation exchange, or other multimodal chromatographic approaches for hydrophobic interaction coupled with AEX or cation exchange. Yeah, there will be a size exclusion. There will be multiple approaches for capturing the protein of interest coming from the harvest, from the upstream process. For these processes, you have to, again, after the first column is step, the expectation is to have about 80% purity for the protein of interest, protein or the biomolecule again, and the optimization of for this downstream process would take place right here, optimizing the chromatographic resin type, resin size. I mean, there are multiple resins for the same mode of chromatography. So that we have to do that, and we have to optimize the flow rate for the chromatography step, ionic strength, and the pH of the media, buffer type, and also the column load. So this depends on the capacity. Probably this is the place where I kind of see it's interesting that the concept that we learn in the chromatography, the column capacity, capacity factor, dynamic binding, static binding, and those principles that we learn, even in undergraduate days, actually coming into play here. So I'm keep going. If you have any questions, please feel free to interrupt me. Otherwise, I'll continue talking. So right here. So what are the testings that we had to do here? And I mean, from right here, we have multiple chromatographic steps. I mean, every chromatographic step add purification. So by the end of maybe three or four chromatographic column steps, we will have very high purity, approaching sometimes 98%, 99%, sometimes more than 95% would be enough. So what we measure here, the protein concentration to measure the yield from each step, and every step add a value to the purification process. So every step gets rid of endotoxins, clearance, so removal of microorganisms, hostile proteins. We have a lot of proteins, then you have to make sure that they are cleared during the process. DNA, we have hostile DNA, we have to clear them. Keep clearance of any components used in the upstream process. For the upstream process, we use a lot of chemicals, just to make sure that the mixing happens. So we use sometimes flocculation agents, we use antifoams, we use even for that matter, amino acids, surfactants. There are multiple species that we use in the upstream process to keep the proteins happy, and also mixing and other upstream conditions under control. So I mean, we have to make sure that anything that we put in the upstream process needs to be cleared during these purification steps. So, and also step yield optimization. All these involve, thousands of testings every time. Like I said, this each optimization, you won't get just in one pass. So this will be doing thousands of tests during optimization and scale up steps for this one. So this happens at much higher scale. So these are these columns are pretty big columns, about 2 to 3 feet in diameter, and in height it'd be again 5-6 feet in height. Okay, so let me go back to this slide. So once we have this one, after the last column is stuff, it goes through multiple step again, like, just to make sure get into the final buffer. It goes through a UFDF step and final get filtered through 0.22 filter. This is just for getting rid of any make sure that all the, if any other microorganism gets through, they all get through won't get through this filter. So when it comes to the final state, this is called bulk drug substance. This bulk drug substance should not have any adventitious agents. So right here we do all the testing for this one, 14 concentration, and the endotoxins, adventitious agents, again, mycoplasma, all the bioburden testing, the protein quality indicating assays, just make sure that we do a lot of testing. These are called release testing that I'll be talking about in coming slides. So, at the, when we get the last step from the downstream process, it's called BDS, bulk drug substance. Bulk drug substance get converted into drug product. So drug product is what happens, like, bulk drug substance get formulation component, excipients, and once we create that and goes into the fill finish line, in the fill and finish, during fill and finish, we fill with the final container process system. This could be a vial or a pre-filled syringe, anything, and that goes into the, then, we do multiple release testing here that I'm going to talk about the release testing. Then this, once the drug product is released, it goes into a stability program. So long-term stability program. This stability program runs usually for up to 5 years or so. And after that, this goes into the storage and cold chain storage, and after that goes into the doctor's office or the patients directly. So let me tell you about, I always talk about the release testing, stability testing. So in IP in process testing, these are the testing that I was talking about during the optimization, upstream process scale up and process development. So we use multiple in process testing. Release testing means, this is the testing that we do to release a kind of, just to tell that, this final DP vial is ready to go to patients. So those are for release testing. And stability testing means, like, we have to make sure that, these are stable for long term, and we keep it in a long-term stability program. Then we have to test in intervals from like 1 month, 3 months, 6 months, the 1 year, 2 year, 3 year, 5 years, something like that. We have a stability time point for each drug lot that we produce. And the product quality would be the testing that we would be like, appearance, color, and clarity testing for the product quality. Then protein concentration, we would use multiple, UV absorbance at 280 from the intrinsic absorbance. And the other side, pH measurement, particulate, moisture, if it is a lyophilized product. Then we use, like, multiple chromatographic testing, HPLC, size exclusion, HPLC, reverse phase, cation exchange and ion exchange. Just to identify the purity and impurity components. We also do something called peptide map and intact mass. Intact mass is by, like, just to show that the molecule that we were targeting to generate have the right mass. So we use intact mass like MOLD or ESI mass spectrometry. Peptide map is just actually digest the protein with trypsin or any enzyme, mostly with trypsin, and make into convert that into multiple peptides. Then those peptides get separated by a reverse phase HPLC and get analyzed by either UV or mass spectrometry. That's called a peptide map. And we use multiple capillary electrophoresis approaches. So this capillary electrophoresis is called zone electrophoresis, isoelectric focusing, SDS page, Western blot, and also SDS page. And let me see. There's a chat. Yeah, there's a question. I will answer that. Fire agents in testing for fire agents in vaccines. So actually, these are the testing, and we also do. In order to do more characterization, do glycan profiles, the proteins. When you produce proteins in mammalian systems and other systems, they get glycosylated. Proteins get glycan. The glycans are more polysaccharide or other monosaccharide chains on their proteins. Those are called glycans. We have to characterize them, and we have to make sure that proteins have disulfide bond. We have to make sure that how many free cysteines are there, because if you have free cysteines, protein can be unstable. And we also have to do high order structure analysis using multiple spectroscopic measurements for circular dichroism, for secondary structure, differential scanning calorimetry, FTIR for both secondary and tertiary structure, DSC. I had this should have been DLS, dynamic light scattering, and fluorescence spectroscopy for tertiary structure. And also we have to measure the formulation component in the final product. For the safety, we have, there are multiple things that we do: residual DNA using picogreen or qPCR, microbial content, multiple methods, mycoplasma, endotoxins, so sterility testing, container process. So there was a question about how do you measure pyrogens in vaccines. Usually pyrogens are measured in terms of endotoxins are a surrogate for pyrogens. So we measure endotoxin use multiple ways. This is called I'm blanking out on this as a name. It's a limulus amibacide lymphocyte, some kind of acid that we use here. It's kind of LAL kinetic test. So this is a plate-based, and this is a plate-based method that we use here. Limulus amibacide lysate. So that's the test, that kind of tongue twister, but that's the test that we use. For the sterility, we do plating. We can do again qPCR. We can do for the sterility while finding microbials. We can do membrane filtration methods. There are multiple methods that we can use here. We can even do novel flow cytometry based method for sterility testing. Container closing integrity is done by helium leak test, and also other areas. The potency is in the efficacy. We have to show that the molecules that we produce are efficacious and effective. So we use ELISA and other immunoassays to show that we have the right biomarkers, and cell-based assays. There are multiple cell-based assays, and also flow cytometry to show that we have the biomarkers and the immunogenicity. Also, we use LCMS for potency, and something called Luminex. It's a way of doing a multiplex assays, that just trying to measure maybe 30 to 50 biomarkers in a single assay using the Luminex instrument. So these are this is kind of no way a exhaustive list of tests that we do, but this is kind of an example of what we do during product release, stability, and during in-process testings. Okay, here's a test, kind of panel of tests that we do during cell and gene therapy testing. I'm not going to go into details. So these are available, at FDA site. So anywhere, we can see that everything is again, same as I said before. We measure the safety, purity, potency, and dosing and identity of the biologic substance. So there are multiple methods that we can use. All these are biologics, typical biologics methods that we use for for example, qPCR based method, ELISA, Western blot, everything is the same, but they have a little bit of complexity the way that we perform this. So again, every raw materials that go into the manufacturing process get highly scrutinized. They are, all of them are get tested before go into the manufacturing process, or at least they make sure that they are good to go. I mean, we have to test all raw materials for purity before using the manufacturing process. We have to make sure that we do the identity test. In this identity test, this identity test could be coming from an NIR, FTIR, NMR, mass spectrometry, and we have to measure the residual solvents, residual heavy metals. I'm sorry, I think this should be here. Residual heavy metals using ICP-MS or ICP-OES, and nitrosamine impurities in raw materials using LCMS. For some, then we have to measure multiple testing, like moisture content from Fisher, other testing, test attributes. Here is an example of what we the tests that we do on the raw materials that go into the biotech products. By this time you should understand why these biologics are so expensive. It's not it's not just that you kind of there's a reason why they are so expensive. I mean, everything. I mean, you can when you think about sub quality, subprime biologics, so they don't do all this testing. I mean, we have quality controls in place, just to make sure that this everything that go into and everything that comes out are safe to use on human. And I just for, and as a scientist in the field, as a at my role, I always tell my the people that work with me, anything that I touch, I should be able to inject into myself, inject into my loved ones. So I mean, so we take a very, very cautious step to make sure. So this method, raw material methods include HPLC, NIR, FTIR, IR. Like I mentioned, there will be NMR, multiple methods that we use to make sure that these raw materials that we use are pure and good to use. Just before, you probably show this. One of the, before I end the presentation, I would like to give you a little bit of my presentation mostly around the technicality, not much of the science, but you can see that there is actually scientific curiosity in this process, but we don't if you look into the process that you are working on, as you look deep enough with your basic understanding of chemistry, chemical concepts, and thermodynamics and kinetics, you will see a lot of science going on there. Like, for example, the entire manufacturing related protein degradation, I mean, most of the protein, the entire industry is governed by one single equation. If you don't believe, if you probably can't believe it, the Arrhenius equation is actually the one that governs in all this stability and force degradation and everything. Because as you can see, every 10 degrees increase in temperature, the reaction rate of a given reaction doubles, right? So you can see that that's why the temperature and the very tight control of these processes are really, really important, especially during manufacturing, storage, that to make sure the quality of these drugs. So I mean, I'm not going to go into much details here. And again, another one, this is why the proteins are highly heterogeneous to lead into multiple degradation pathways, including, the unfolding, aggregation, and there will be multiple pathways that are governed by thermodynamics and kinetics processes. So we have to, just take into account and all these and understand the processes. Even I mean, especially as chemists, we are the one who behind these kind of an understanding these processes. Probably the biologists won't get. I mean, we work with immunologists, biologists, cell biologists, molecular biologists, but we are we are probably in the center of everything. So we understand molecular biology. We understand the molecular biology and every aspect of it, and also since we understand the thermodynamics and kinetics, then we are actually in a better place give more guidance and developing and scaling up these processes. Okay, and okay, I think that's I would like to leave it at this point with some questions here, and the idea is just when we come to the mechanistic side of it, like protein degradation. I mean, there'll be like lot more to learn here. There'll be like there's a lot of information out there, and even if you YouTube it, there are multiple sources that you can get this information. But I'm happy to continue these discussions and this series, or like kind of convert this into more biopharmaceutical manufacturing related curriculum at Peradeniya or somewhere wherever they are interested. And just there are a lot more to learn, and I'm willing to give more seminars around more the techniques, or around more protein biochemistry, the mechanism, and the degradation pathways. There are multiple things that we can talk about. But I want to go into the next slide, where I was asked to give a little bit of who I am, how I build, how I accumulate knowledge and experience. So I can tell you the cornerstone of my, I would say, to get here. Instead of saying success, to get here, so get my biopharma expertise, it came from both from analytical chemistry background, where I got into the best thing I did in my life. I was like thinking about doing chemistry special, even when I was at even before I started my A level. So that was the inspiration there. Biostatistics. I mean, that's one of the best decisions that I made. Came into Peradeniya, did my biostatistics as minor subject, and biophysical chemistry. I mean, I got a lot of research and thorough understanding of biophysical chemistry at KU when I did my PhD, and biology. I mean, this is kind of a lifelong event. Like from A level to Peradeniya to KU, I got exposure to even I kept learning. And I mean, biology and all these are the cornerstone of kind of getting in here. But you don't have to do all of them, but just you have to find your niche, what you are good at, and just try to build on that. That's the thing, the building on that. I started Peradeniya, went to KU, and I started as one of the most innovative companies. And I mean, my career has been blessed with working on most advanced products. The Dendrian was the first company to commercialize a cell and immunotherapy, and went into Armagen to work on blood-brain barrier crossing therapeutics. Here is more vaccines. At Bristol Myers Squibb, I got to work on multiple biologics products, and at Partner Therapeutics worked on cytokines, and at my current company, I worked on Professor Anthony mentioned that work on multiple vaccines that are going into to bacterial space caused by antibiotic resistant species, multiple pneumonia, and we are kind of working on so this is the current job is most the frontier in the synthetic biology. So we are one of the most advanced companies in the world doing synthetic biology, and we are in a very, very advanced stage in the vaccine development. So that's that. And so building expertise and expanding networks, you all should do that. Expanding it. I mean, it's the thing is when I did my DS, special degree in chemistry. I mean, I have choosing analytical stream. I mean, I chose the analytical stream, attending, but we had the option to choose analytical or biochemistry. I chose analytical stream, but attended all the biochemistry lectures. that hit it. So then also like I said, biotech, biostatistics. So that's one of the best decisions. Even though I do, I mean, we are chemists. I mean, right now, probably 70% of my work is supported by biostatistics. So, and also, thanks to Professor Kumar, Professor Liyanage, they all did a great awesome job in carbohydrate chemistry. So our Vaxcyte programs run on polysaccharide. Actually, we just produce polysaccharides from using bacteria and conjugate them into proteins. So this is whole thing is polysaccharide, and again networking. So I was the president, secretary, treasurer in Chemical Society, and also had the luxury of working with Professor Namal, and I thought that helped a lot. And thermodynamics and kinetics that I learned during my undergraduate days, and also again expanded during my graduate work. So this is kind of keep me in the spotlight. So, and again, the strong spectroscopic techniques. So spectroscopy is really, really strong at the chemistry program. So overall raw material programs depend on spectroscopic techniques. So just raw materials, everything depends on, the characterization of even the APIs, drug product, everything depends on spectroscopy. So, okay, I can say that, one thing if I didn't do well, it's like, skipping solid state chemistry, I wouldn't recommend it. So during the PhD, again, I got exposure to, this is where actually the pivot point came into my life from having that chemistry experience and doing recombinant DNA technology, protein purifications during my undergraduate work, during my graduate days. So biomolecular conjugation, getting exposure to fluorescence spectroscopic techniques for single molecule detection, protein dynamics, and also had the luxury of developing novel analytical methods for protein ligand binding, and also again, like I said, thermodynamics and kinetics. Also expanded my horizon, understanding the biomolecular world. Again, like I said, also I built a relationship with external lab that ended up getting me to work on vaccine. So this is where the pivot point happened. When I did a lot of kind of molecular biology work, protein work, that's the thing. You probably, this is not when I came to the United States, I was thinking about my experience was working on environmental science and just doing some sort of things. But you have to keep your mind open, just be perceptive to what's in the path and the change. You have to look beyond today, you have to look into 10 years from now and what's going to be there. Just try to grab that skills. At least you should have transferable skills. When I came to from the chemistry program from Peradeniya, I had transferable skills that actually could have used in the world of biotechnology. So that's how you kind of thrive. You just don't want to survive, you want to thrive in this world. So I think with that, I would like to answer any questions, just talk about, the career development, anything, you name it. And also, please feel free to reach out to me. I'm on LinkedIn, and I am apparently, I know some of you are from Facebook too. And also you probably can get my email. So with that, I would wrap up this webinar and hoping to contribute more in the future. Thank you so much for your patience, attention, and apologize to you because my time is still 5:40 a.m. So I started this. I woke up at 4 o'clock, so I may not have been as sharp as I would have been in the daytime, but I survived. So thank you, Professor Anthi and Professor Manav-Devi, for this very critical work to inspire the young crowd and this budding scientist. I really appreciate it. I have a lot of respect for you guys for that. And if you need any help, I'm well, acquainted with the scientific community here, and if you need any help, different aspect, giving a talk or someone I can talk to people here, and you will be. Again, thank you so much for doing this, and all the young scientists. I am happy to help you in any way I could, and keep learning and networking. It's you only have to be good enough at every point of life. Just going from you don't have to be like the smartest one in the class. You don't have to be good enough, but the rest is who you know, what you know, and what you want to do, and your inspiration. And there's a joke, and if you can find inspiration, get a bank loan and buy a house. Inspiration will be with you every day. So with that, I would stop.
Thank you very much, Dr. Roshan Liyanage, right early in the morning in San Francisco, United States. And it was a very informative and highly motivational and also very exciting talk. So this is, with that, we would like to have some questions from the audience. You can use raise hand option available in the Zoom.
Oh, can I ask a question?
Sure,
okay.
sir.
Okay, this is Ratnaka Bandara. I'm happy and proud to see where you are now. Actually, I remember you as an out-of-the-box thinking student those days. Is that correct? Yeah, that's how I remember you. Yeah.
Thank you. I have to give you a big thank to who I am today. So actually, you probably don't remember this. You probably don't remember this, but one day I was working on the Buddhist Society. We had a Peradeniya, and I was working. I skipped one of your classes to do some work to get the period organized, and I went to Kandy and came back, and I came back and I lied to you. And you recognized that, and you told me that I know that you're lying, and just as
you lied.
and as you can do that. Actually, that's the day actually changed my life. I became a better scientist because of that advice. I never lied after that.
Okay, good to hear that, Roshan. Anyway, your presentation today on biopharmaceutical products is very comprehensive, and I learned a lot today, right? So now from your presentation, it's very clear that to get a product to the market, biopharmaceutical product to the market, you have to have a battery of tests and then clinical trials and so on, which means it takes a great deal of time, and a lot of time, right?
Yes.
Then my question is, however, COVID-19 vaccines were developed within a short period of time, right? So how did they do that? Or did they cut short some steps, or how did they do that?
They didn't. I mean, just to be fair. So this the COVID vaccines came into the vaccine came into the market in less than 2 years, 1.5 years or so.
Yeah.
But the research went into the mRNA and nanoparticles. The mRNA actually started about 50 years ago. If we so the funding from the U.S. government for 5 decades actually helped brought them to the market. So I think we underestimate the power of the basic research. So that's the problem. People don't understand and don't have the interest to get into the depth of it. So research started about 50 years ago, and also the mRNA was in clinical trials for like immunotherapy treating cancer over few, 5 to 10 years, it was in clinical trials. So during those clinical trials, the industry had the opportunity to assess the safety, the toxicity, and other aspect of it. So the other things like you can just create an mRNA molecule you can design an mRNA molecule in two weeks, but like you said, you have to do multiple testings, but these testings have been done on multiple areas. Even for the lipid nanoparticle systems have been developed by Bob Langer at MIT, have been in the research, they've been the toxicity, especially the safety, have assessed for a very long time. So anything we always have the trade-off, right? The long-term effect. I can't speak to that because long-term effects could be we don't know. First we have to live to see that. So I think I don't believe that there have been they have cut corners or anything like that. But there is enough knowledge about the toxicity, safety, and efficacy of these new novel vaccines. So I don't have any hesitation to take them.
So it's quite safe, yeah?
It's quite safe.
Okay, thank you, Roshan.
Yeah.
Okay, great to listen to you.
Yeah, nice to talk to you, Dr. Ratnayaka Bandara, Professor.
Okay.
So Roshan, may I add few words?
Sure.
So I'm very glad that you presented the very nice details of all new things. And also it took my memory down the past because actually at that time, analytical chemistry and biochemistry, we had both streams, but unfortunately we couldn't offer both at the same time. So therefore, actually we couldn't do that lots of biochemistry. I feel like even now, if we have more biochemistry into our curriculum, would be helpful. So when you
absolutely,
yeah,
absolutely.
So yes, and also in the future, maybe we will invite you because we have some. We are going to have a biomedical science program at the PGIS MSc program. So
okay, right.
Workshops on pharmaceuticals, maybe technology. That side we can invite you. And yes, also I want to ask you one question about the presentation. So the gene therapy is in effect in US now. I mean, it is used for treatment for patients.
Not many ailments, but it's been approved for few diseases. Just treating some diseases around eyes, and it's been approved for a handful of diseases at this point.
I see. Okay. Right. Thank you very much, Roshan.
Say hi to Professor Nama for me.
Sure, I will. Today is not home. Thank you.
Okay. All right.
Thank you, madam. I think Swarna madam has a question. Swarna madam.
Dr. Roshan, it's a very interesting talk, and I just wanted to ask you about this protein carbohydrate conjugation. When it comes to the bacterial polysaccharides, they do have a repeating unit, and so the part that is looking out is the active component, right?
Yeah, so we are kind of into the immunogenic component.
Madam, are you with us?
Yeah. Can you hear? It's not clear?
Now it's clear, madam. Madam?
I think there is some problem with the audio.
You use it. I will put it in the chat.
Yeah, okay.
I think Madam experiencing some technical difficulties. Madam?
Yes, she was
I'm
trying to
typing.
type it.
Until such time you can get someone else's question.
Yeah, you can ask anything.
Me, sir. I hope I'm audible.
Yeah, I can hear you.
Sir, what are these opportunities, like what are the availability of these opportunities like for students who are not in like doing science undergrads or graduate study, like if they're like not directly studying anything related to this, what are the opportunities like for students like that?
So, okay, so opportunities would be in, it depends, if the students they are already in the United States, it's a different story. But if they're in Sri Lanka, are you talking about they do not follow graduate studies, like someone who came out of A-level or something like that?
No, so like say if they're not studying analytical chemistry, but they're studying like another stream, science related stream, but they want to get into this field, what is the opportunity like for them?
So, I mean, let me tell you about something real quick. That for example, if you have a mathematic backgrounds or statistic background, there'll be a lot of use in this field. Like for the clinical trials, we use a lot of statistics. Just for the data analysis part of it. And if you have a computational background, for computational background, we use a lot of computational modeling of the proteins and other biomolecules. Those are the areas that others can focus on. For if you have any exposure to microbiology, anything like that. So if they don't have the experience, they want to get into the field. So either for the getting into biotechnology, it would be like, you have to probably get exposure to some sort of science, maybe immunology, cell biology, microbiology. If you don't have it, just get it. Just go do like master's degree or maybe a certificate programs. There are multiple ways to do that. Even there are a lot of free resources right now for like Coursera. It's really cheap. You can do courses from MIT, Stanford, Berkeley, or any university in the United States for really, really cheap. And LinkedIn has great learning resources. So I think, those are the opportunities and avenues they can follow. But I would, if you are in Sri Lanka, I would definitely suggest that, maybe doing an MSc or there is other programs. I think Colombo University has molecular biology departments. It's very, very strong, as I know it. So they have multiple graduate, undergraduate, or graduate programs or certificate courses. I think, there are multiple places that you can get that experience.
Thank you so much, sir, for that answer. Thank you very much.
Yeah, no problem.
Dr. Roshan, I think Madam.
I have the question now. It says, how would you conjugate the protein to the polysaccharide, keeping its biological activity, and it's going to be a specific So we have, for the conjugation, we have a specific proprietary method that we, during the protein expression, we use a protein called ECRIM, but we have like cell-free protein expression system. We also include non-natural amino acid, depending on the type of vaccines. We incorporate 4 to 6 non-natural amino acid, use the CDAP chemistry to activate those non-natural amino acids. They have an azido group. Those azido groups get activated, and we do the polysaccharide conjugation specifically to those non-natural amino acids. That way, like we keep the like this kind of epitope of the carrier protein open for T cells, so that way we create both antibodies and memory T cells. The second question was question: is it effective to use polysaccharide as it is, or an polysaccharide with several repeating units? So there are currently Merck has 23-valent formulation using just the polysaccharides for pneumonia. The problem with just using the polysaccharides, they are not very immunogenic. So you get some response, you get some protection, but the memory, they won't create memory T cells. So that's the durability is in question. So that's the reason why we use this conjugation approach. You can't you can use them, but they are not highly immunogenic.
Thanks. Yeah, I understood. Okay, thank you very much.
Thank you very much, madam, and thank you very much, Dr. Roshan Liyanage. Since we are running out of time, we would like to end with the question and answer session. If there are more questions, I'm pretty sure that you will have more and more questions for Dr. Roshan Liyanage. You can send them to Dr. Roshan Liyanage. So thank you very much once again. I would like to move to Akalanka. Akalanka.
As this evening come to a close, the 14th webinar of the Inspire session has come to a successful conclusion. It is with great pleasure I present a vote of thanks on behalf of the Department of Chemistry of University of Peradeniya and members of the Inspire team. First, I would like to express my sincere gratitude to Dr. Roshan Liyanage for accepting the invitation and conducting an instructive webinar to share his knowledge and experience in analytical chemistry from the perspective of frontiers in biotechnology. Further, it was an amazing experience to gain insights into the latest developments in the field of biotechnology. This is treatment, prevention, and novel biopharmaceutical modalities. Dr. Roshan Liyanage, we thank you very much for inspirational presence on the Inspire stage. Next, I would like to convey my sincere gratitude to Professor Ayanti Navaratna for inviting and hosting the guest speaker on the Inspire stage. Furthermore, I would like to extend my gratitude to Professor Manavadevi Ganahenage, Head of the Department of the Chemistry, for her encouragement, guidance, and support to webinar a success. In addition, I would like to thank the attendees for joining us this evening. Your presence has been a source of motivation and support for us. I'm confident you would have found this webinar a source of inspiration to aspire to join the community of scientific scholars. Finally, I would like to invite all of you to extend your support by participating in the next webinar. Yours is the earth and everything that's in it, which will be conducted by our invited speaker, Dr. Mudita D. Senratyapa, who is the head of the John Keel's Research and Vice President at the John Keel's Holdings, Sri Lanka. This webinar will be held on 22nd of December at 7 p.m. Dear Dr. Roshan Liyanage, you are invited to join for the Meet the Speaker session using the other link provided. Thank you. Have a pleasant evening.